Cadherins are an important component of cell adhesion. The boundaries between tissues are created by different cell types, and many of these different types and amounts of cell adhesion molecules are there, including cadherins. And there are different types of cadherins, different amounts of cadherins that relate to how much certain cells adhere to one another. But these are the major cell adhesion molecules that are involved in development and involved in holding our cells together once we become adult organisms. Cadherins are transmembrane proteins that interact with the cadherins from other cells, and we can see that in this illustration here, where the cadherin goes across the cell membrane, and it has 5 subunits which are all calcium binding. They have a calcium binding site. In fact, it is calcium-dependent adherin. That's how it got its name, calcium adhesion. And you can see that this cadherin molecule then, this is on the outside of the cell, here's the cell membrane, will extend through the cell membrane. There are some phosphorylation sites, and it hooks to the cytoskeleton via what are called catenins. Phosphorylated regulation sites relate the catenins to actin and to the cell membrane per se. This illustrates now that you have a cell here and a cell here. These cadherin to cadherin binding areas then indeed are what keep the cells together. Let's go through part B of the slide here now. In this case, the cadherins between adjacent cells have been broken down by adding some antisense oligonucleotide. And so instead of the cells of this amphibian embryo sticking together, basically they are just randomly positioned within this epidermis. Here is the normal control in which the cadherin-mediated adhesion is normal, and you can see that the embryo is developing in a normal fashion. So just to kind of review this slide, multiple functions of cadherins then. One function, of course, is to cause the cells to adhere together. Also, they are associated with actin filaments, and what they do is to stimulate the polymerization of actin into filamentous actin. In other words, G-actin into the filamentous state. And that allows cells not only to adhere together, but to move and to migrate, which is an important component in development, because development is more than just differentiation. It's also getting the cells to the right place at the right time. Now, it's important that we consider the amount of cadherin as well as the type of cadherins for correct morphogenesis to take place. And what this is doing is showing you a graph in which surface cadherins can be increased in different kinds of tissues, and that causes those different kinds of tissues to aggregate to a stronger or to a lesser degree. Here we see cells that have been mixed together for 4 hours, and the green staining cells are cells that have 2.5 times as much cadherin per cell surface as the red cells. And so initially at 4 hours they're basically intermixed, but after 24 hours then, the green staining cells, which have, like I say, about 2.5 times the amount of cadherin, will be more tightly bound together, so that they form the inner portion of this tissue as compared to the outer cells, which have a lesser amount of cadherin on their surfaces. And so this is really how cells and tissues form in the body and how they form relative to each other and why the right components get to the right place. Let's look at another example of neurotype cadherin for correct morphogenesis. There are several different types of cadherins. Of course, we just talked about quantity being important, but certainly the type of cadherin is important as well. There are E-cadherins, which are found in all early embryonic cells of the mammal. That's shown here, the E-cadherins. This is the neural tube area, and these E-cadherins are found in this early embryonic ectodermal epidermal tissue. P-cadherins are cadherins that actually anchor the placenta to the uterus during pregnancy. So that's present very, very early in the developmental process. The N-cadherins refer to neural type cadherins. They're associated with neural tissue. Here we see a neural trough that's going to give rise to a neural tube in this organism right here. and you can see that the end cadherins are in highest concentration in that neural area. There also are cadherins, for example, in the retina of the eye, and there are protocadherins, which is a class of cadherins that lack attachment to actin, and through the catenins they simply don't interact. But let's now look at another type of tissue here relative to the end cadherins. Let's look at another type of cadherin that's present. This is again an extension of the end cadherin that we saw here, but this is in the zebrafish. Here's a normal zebrafish. Here's the neural tube forming, neural trough and neural tube, and you can see that the end cadherin is very prominently stained. And in this case, though, this is a mutation in which end cadherin is not present, and you see that there's an abnormal structure in this neural tube, and it's not really forming correctly. So if you look at this diagrammatically, the end cadherin would be in this neural tube area, the neural crest area. The E cadherin, the embryonic cadherin, would be in this location, and that's how normal development takes place, to have the right cadherins in the right place. So not only does it require the normal quantities of cadherins, but also the normal type of cadherins for the normal morphogenesis. And this stems from the time of implantation of the embryo until the formation of the adult organs. Here's an example of a frog gastrula in which the cadherin adhesion and morphogenetic movements have been interfered with by taking and putting a non-functioning end cadherin gene into this case the right side of the frog embryo here, and you can see that the neural tissue is sort of not really formed in an appropriate way, and here we have neural tissue on this half of the embryo that is perfectly normal, and it basically is forming in the correct manner, as well as the cadherins on this side showing the endodermal area as well as showing some mesodermal tissue. In this cross section, this is stained for F-actin. Now that's filamentous actin. Remember we said that cadherin will cause and stimulate the formation of F-actin in certain cells they're associated with, in fact, most all embryonic cells. And here, in this case, the red staining cells are the result of notochordal tissue being affected by a morpholino that's been added to this notochord material. And instead of the green stain, you get a red stain, which is the morpholino, but it's eliminated the formation of the actin into filaments. So here we see that filamentous actin is stained green in all these other areas, but where this morpholino has made it so that the end cadherin mRNA cannot be translated, in this case then we do not get F-actin. Clearly you don't get a normal neural tube at all forming. The other thing that I wanted to mention relative to the cadherins is cell migration. I mentioned that, certainly we have to have differentiation, but we also have to get cells to the right place. And so the cadherins help to do that. They help cells adhere together, but they also stimulate the formation of actin filaments so that these cells can move along extracellular matrix channels and end up where they're supposed to be. If they're neural crest cells, they end up going down into the gut and forming nerve cells, as well as going down into the epidermis and forming pigment cells by this migration ability. And that migration ability is allowed by, again, cadherins relative to actin filaments in the cytoskeleton that these cadherins have stimulated to form. So the initial stage of this, here we can see a cell that's now become polarized. The cadherins have affected polarization in the sense that they've caused actin filaments, that's indicated with a green stain here, to form at this, what's going to become the leading edge of the cell, and this will be the trailing edge of the cell. In a more advanced state, again, you have the actin filaments in this area and the external portion of the cell at the leading edge, but these are actin filaments, actin cables that have formed relative to that cadherin. And so the cell is migrating in this direction. This is the leading edge. This is the trailing edge. And the other part of this is, after the polarization, then you get an adhesion in the center of the cell, adhesion to that extracellular matrix, and finally there's a release of adhesion in the rear of the cell, and all of these are coordinated by the cadherins. the adhesion, the release, and the forward migration. So, if we are to look at the development of tissues in general, we have to realize that for things to get in the right place at the right time, for something to become a finger bone and not a pelvic bone, or to become a retina and not a corneum in the eye, we have polarization, and this polarization is a result of differential cell migration. There are sorting movements then that get the cells to the right place in the right amount of time and get it there so that inductive interactions can take place and cause these cells to differentiate in a certain direction. All of these developmental processes then require the cell-to-cell communication and adhesions from cadherins and from the cytoskeleton.